Hello everyone, myself Dr. Vivek Kumar, Professor Himalayan School of Biosciences, Swami Rama Himalayan University, Jolly Grant, Dehradun. I am again with you with another topic on endophytes, that is endophytes and host plant growth. Endophytes, the endophytic microorganisms, they cause some positive benefits on the host plant which leads to the improved growth and yield of the plant. It has been observed that long-term approaches of organic and inorganic fertilizers have been used beside pesticides required for the enhancing crop production. So these applications negatively influence the soil quality. They negatively influence the human health also, and they also cause the environmental pollution. So to minimize this detrimental effect of these inorganic chemicals, innovative methods based on microbial inoculations are being gaining more attention, and these microbial inoculation, nowadays people are using endophytes to inoculate the plants. So plants and microbes, they form a symbiotic association which benefit both the partners. The plant, they provide protection and nutrition to the microbes. On the other hand, whatever the secondary metabolites, beneficial chemicals produced by the microbes, they enhance the plant growth. So more importantly, plant microbe symbiosis influence on host plant growth and health, which effectively ameliorates the agricultural traits and improves soil quality and nutrient cycling. So plant growth promoting endophytes, they inhabit plant tissues, we have studied before, and which facilitate nutrient exchange and enzymatic activities. So the plant growth promotion by endophytic bacteria, the endophytic bacteria, they utilize the same mechanism which is employed by the rhizobacteria. Whatever the mechanism employed by rhizobacteria in enhancing the plant growth, the same mechanism is being employed by the endophytic microorganisms. So that promotion by the endophytic bacteria on a host plant, it could be direct or indirect. Now these are some indirect benefits, these are the direct benefits. The indirect benefits for the suppressing plant disease by production of antibiotics, ammonia, induction of plant systematic resistance, protection against plants, pests and herbivores, phytoremediation by removing heavy metals and hydrocarbons. The direct effect could be production of phytohormones like auxin, cytokines, gibberellin, etc., elevating abiotic stress, draws, salinity, etc. Biofertilization means that the endophytes, they provide benefits in the form of nitrogen fixation, phosphate solubilization, etc., and plant nutrient availability. In direct plant promotion, let us discuss first the phytohormone production. So plant growth, these are the plant growth regulators or the phytohormones. They are the organic substances that modify, inhibit or promote plant growth and development even at low concentration, less than 1 millimolar. For agricultural applications, they are either chemically synthesized, extracted from plant materials or produced by microbial fermentation. The main phytohormones produced by endophytic bacteria are auxins, cytokines, abscisic acid, ethylene, etc. Indole 3-acetic acid or IAA, it primarily promotes plant cell elongation and cell differentiation. In the process of symbiosis between the bacteria and plant, the IAA, it stimulates adventitious and lateral root development. It promotes nutrient access, enhances root exudation to increase the interaction with the soil microorganisms. The common endophytic indole producers, they are azospirillum, azotobacter, alkaligenes, arbospirillum, enterobacter, etc., etc. So these are the bacteria. Other than these bacteria, there are most of the bacteria, they are able to produce the indole acetic acid. So plant defense hormone, another, this is ethylene, which is involved in stress response as well as the developmental process, including root development. In rhizobia legume symbiosis, this ethylene participates in the nodulation process also. So bacterial endophytes can possess 1 aminocyclopropane, 1 carboxylate deaminase, ACC deaminase an enzyme which generates the nitrogen source ammonia and alpha ketobutyrate from the ethylene precursor. The endophytes, they can promote plant growth under nitrogen limitation conditions through the secretion of ACC deaminases. Now several studies reported endophytic bacterial species which produce another hormones, gibberellins and cytokines. The gibberellins control plant growth and development through enhanced seed germination, stem and leaf growth promotion, stimulation of flowering and fruit development, and delaying plant aging. Cytokinin, on the other hand, controls cell division, cell differentiation, increase resistance to biotic and abiotic stresses, enhance phloem transport, promotes flowering and auxiliary bud formation. So these hormones, indole acid, cytokines, gibberellins produced by the microorganisms, so they all cause a benefit effect on the host plant. Now the picture shows that these are the direct plant growth promotion, like the phytohormone like oxygen ethylines, then volatiles and other phytostimulators, and the nutrient availability, your phosphate, your potassium, your nitrogen. So these are the direct plant growth. Indirect means pathogen protection using your antibiotic production, SN production, quorum quenching, induced systemic resistance. They also manage abiotic stresses. So we can say that endophytes, once they are inside the host plant, then they benefit the host plant by direct and indirect means. In one study, endophytic azoospermillum lipophilum, it was inoculated in a maize plant treated previously with gibberellin inhibitor synthesis and then subjected to the drought stress. The inoculated maize plant performed better than the uninoculated control, particularly under stress due to the bacterial gibberellin, because the azoospermillum, it produced the gibberellin. So modulating plant indoleacetic pools is an important trait by which endophytic bacteria, they can enhance the plant growth. So indoleacetic production by endophytic bacteria can result in improved plant root biomass, improved surface area, increased production of lateral roots and in the host plant. In one study, culture supernatant bacteria stimulated root formation in kidney beans by increasing root length, number of developing roots, because that culture supernatant bacteria contains phytohormones. So this shows that in culture supernatant, the hormone indole secreted by the bacteria showed a part in the development of plant root system. Another evidence of the role of bacterial indole by the Pseudomonas putida, which are defective in IA synthesis. So inoculation of this strain was unable to increase plant root growth and lateral root formation. It means that when the bacteria was not able to synthesize hormones, that was not able to enhance the root growth and plant growth. On the other hand, when the bacteria was able to produce phytohormones, it was able to enhance the plant growth. Another, nitrogen fixation. The manufacturing nitrogenous fertilizers is costly, we understand that, and their usage cause lot of environmental side effects. So biological nitrogen fixation process by nitrogen fixation by bacteria, it accounts for about 2/3 of the globally fixed nitrogen. So apart from photosynthesis, the biological nitrogen fixation is the most remarkable biological process, and this process is limited to prokaryotic microorganisms only, for example your bacteria. So bacterial endophytes, they can establish close relations with certain crops. So plant endosphere contain excess carbon and lack of oxygen, which presents or permits suitable conditions for nitrogen fixation that can be transported by endophyte to the host plant. So the endophytic bacteria, they can provide nitrogen, they can fix nitrogen without forming root nodules, as in case of Rhizobium legume symbiosis. So some endophytic bacteria possess BNF genes, nitrogen fixation genes, that enable them to convert nitrogen gas into the nitrogen form, which is taken up by the plants or utilized by the plant. In Brazil, in one study, it was observed that sugarcane plant cultivated without nitrogen fertilizers and infected with diazotrophic endophyte. Diazotrophic endophytes means those endophytes which are able to fix atmospheric nitrogen, such as Herbasphaerum, Ceropidaceae and Acetobacter diazotrophicus were able to derive their nitrogen needs from atmosphere. Some endophytic strains of Enterobacter and Klebsiella have been reported to fix nitrogen and promote growth of sugarcane under notobiotic and natural conditions. Now, another feature of this endophyte, phosphate solubilization. The plant take up the phosphate in the form of H2PO4 and FHPO4. So 95 to 99% soil phosphorus is non-available for the plants, because when we apply soil, what is the phosphorus present in the soil, or farmers apply phosphate fertilizer in soil, it gets fixed in the soil. and it becomes immobile, insoluble. So in agriculture, the phosphate deficiency is compensated by applying chemical or organic phosphatic fertilizers. So endophytes, they increase phosphorus availability for the plants as they dissolve insoluble phosphate by secretion of organic acid or chelation or using the ion exchange process. Furthermore, the endophytes can secrete phosphate solubilizing enzymes such as phosphatases, phytase and CP lipase. Secretion of organic acids such as citric, malonic, fumaric, tartaric, gluconic, acetic and other, they are considered in the main mechanism behind the phosphate solubilization. So when the organic acids are produced, the pH of the environment goes down and the phosphate is solubilized. So pH solubilizing activity of the bacteria is usually correlated with the decrease in pH value of the medium. Now these are the mechanism utilized by the microbes to solubilize phosphate. Enzyme is there, we have discussed. Organic acid phosphate assimilation is there, exopolypyrrolate secretion, and then mineralization, chelation, and then holds free form of insoluble phosphate. And when they hold this form of phosphate, they are provided to the plant. We can understand one thing, that phosphate or other minerals are available or uptaken by the plant only if they are in the soluble form. The phosphate solubilizing bacillus species, they secrete itaconic, lactic, isobutyric, isovaleric and acetic acid, and thus acidifies the medium and digestate during the process of symbiosis. Other pH solubilizing mechanisms are the secretion of exopolysaccharides. Another mechanism could be the degradation of P containing substances. When they are degraded, phosphate is released. When it is released, it is soluble and is taken up by the plant. Another mechanism could be the siderophore production. The siderophores, they are low molecular weight organic compounds, iron chelating compounds, and we understand that iron is essential for all life and is required as a cofactor of many essential enzymes, involved in many biochemical processes, involved in nitrogen fixation, and in most soil iron is not available for plant absorption because it is present in the insoluble ferric form. Other forms associated with the other chemicals. Under iron deficiency, many microbes, they can produce and secrete low molecular weight organic compound known as siderophores, which binds to the iron, ferric or ferrous form, and are divalent metal ions which are essential for the plants. So besides the delivery to the plants, siderophores also participate in scavenging undesired metals ions in the rhizosphere to prevent uptake by the roots. These includes these metals, zinc, cadmium, chromium, etc. etc. Siderophores can also stimulate the plant-induced systematic resistance response to elevate the toxicity of trace matter to the plant growth. This picture shows how the endophytes, they benefit the plant. They control the pathogens, they provide nutrients, they solubilize phosphate, they fix atmospheric nitrogen, they provide induced systemic resistance, they produce phytohormones, siderophores, and they manage the abiotic and biotic stresses. Normally the endophytic bacteria produce siderophores only in soils with iron limitations. It has been shown that siderophore produced by endophytic bacillus strains which promoted the growth of common bean under iron limitations. The siderophores of the bacterial endophytes also outcompete phytopathogens. How? When the phytopathogens, they also require iron. So when the beneficial bacteria are there, they produce siderophores, they remove the iron. Once they remove the iron, this iron is not available to the phytopathogens. So in this process, the phytopathogens, they are outcompeted. In another research work, citrus endophyte methylobacterium, they release hydroxymate type of siderophores into the medium. When the siderophore containing supernatant was applied to xylella, it promoted the growth and chlorophyll content in the plant. Similar results were reported for various symbiotic interactions with different agriculturally relevant hosts in greenhouse experiments. So this picture again shows that the left hand side there is no inoculation, right hand side there is inoculation of the plant with the endophytic microorganisms. When the microbes are associated, they enhance the plant growth, they provide protection against biotic stress and abiotic stresses. Finally, the keywords, almost every plant species is found to be in association with the endophytes. We can say that endophytes, they are present in all type of plants, in all locations, and these are the promising agents. Endophytes are promising agents that promote their host growth even in stress environment. It could be biotic stress, it could be abiotic stress. They also play a role in phytoremediation also. They flourish, they enhance their host growth either directly or indirectly. These are the potential sources of sustainable agriculture. If we apply these endophytes as a biofertilizer to their potential plants, then obviously we are going to reduce certain amount of chemical fertilizers, we are going to improve the soil fertility, and we are going to improve the yield in a safe manner, in an organically cultivated manner. Instead of using agrochemicals, which are the main cause of soil health and environment depletion, the farmers should start using endophytes as the biofertilizer. They can enhance the plant growth, they can prevent the phytopathogens, and they will ultimately increase the yield of the growth by maintaining the sustainability of the environment. Thank you so much.